Welcome to the Performance Around the Clock podcast. I am Professor Satchin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock. And I'm super excited to have as my guest Professor Susan Golden, who is also one of my scientific heroes. Professor Golden is a professor at the University of California, San Diego, and also she has been, and just recently stepped down, as the executive director for the Center for Circadian Biology at UC San Diego. And she has been elected to the National Academy of Science. That's one of the highest honors for any scientist on this planet, to say. And she was also HHMI, Howard Hughes Medical Investigator, Professor, for a while. And during that time, she really transformed circadian rhythm education. So welcome, Susan.
Thank you. It's a real pleasure to be here.
Yeah, so you worked on cyanobacteria, blue-green algae. Literally, the word cyan means blue-green, and yes, unfortunately, it was called algae for a very long time. And this particular bacteria, cyanobacteria, has given us a lot of fundamental principles in both photosynthesis, or how plants split water and make carbohydrate, to say, and energy, and also many of the fundamental principles in circadian rhythm. These days, there are very few people working on cyanobacteria, and when we think about biomedical research or biological research, we always get excited about curing cancer and stem cells, etc. How did you get into cyanobacteria research?
I started out working on cyanobacteria not in circadian research, but it was really a matter of opportunism of what laboratories were available when I was a graduate student, and I wanted to work in an area of gene regulation. I'm pretty old, and molecular biology was pretty young at that time, and I had the opportunity to work in the lab of Lewis Sherman, who at that time was at the University of Missouri, Columbia, where I did my PhD, and he had just come back from a sabbatical where he had brought back from a lab in Europe a transformable cyanobacterium, and it was Synechococcus elongatus, at least what we now know is Synechococcus elongatus. And my job as a graduate student was to develop a genetic system, cloning vectors, and work out transformation protocols with the goal of identifying the genes and proteins that were responsible for making up photosystem 2. Okay. And I didn't discover that, but I was at the right place at the right time, and in fact, the genes that I identified, one of the genes that I started working with that I found as a herbicide-resistant mutant, it did turn out to code for one of the most important proteins on the planet, which is the reaction center D1 protein of photosystem 2, which is critical for splitting water, making oxygen, and making oxygenic photosynthesis work.
So then let's unpack some of the terminologies here.
Yes.
Because first, let's go back to cyanobacteria and why this is such an important organism. So you always say that cyanobacteria are the oldest fossils, and cyanobacteria, this class of organisms, are also some of the oldest ones.
They are absolutely some of the oldest organisms on Earth, and some of them that grow in aggregate in a way that they have been fossilized. And so, in fact, the oldest fossils are fossils of cyanobacteria. So they have really been around as far back as anyone can date life. And probably not the first organisms on the planet, because there are other photosynthetic microorganisms that have simpler photosynthesis systems, and they probably predated cyanobacteria, but they really go far back, and they are the instruments of the oxygenation of our atmosphere.
So they go back 3.5 billion years?
Yes, yes.
3.5
really far.
Yeah. So when there was no other, so they're bacteria. So that means
they are bacteria. They are absolute bona fide bacteria. Yeah.
So there was not even a single cell with nucleus or mitochondria when cyanobacteria started.
There are, yes, even, and to this day, none of them have have any of those things. Yes.
And the other thing that you said was photosystem or photosynthesis. And on this, since these bacteria were almost the ancient ones that we can think of now, and there was nothing else, so they could not find another animal to eat on, or so they had to be completely self-sufficient, right?
Absolutely.
So that means they had to make all the essential elements of life. And when it comes to all the essential elements, it boils down to, I think, two fundamental processes. One is photosynthesis.
Absolutely, photosynthesis, which is a way of sunlight is the one thing that we have in abundance, and so a great source of energy, and then carbon dioxide from the environment. And so the cyanobacteria evolutionarily are the precursors of the chloroplasts of plants that let plants carry out that process. And cyanobacteria are like free-living chloroplasts, and they, in fact, the process in plants is the photosynthesis that was invented by cyanobacteria. But there are other bacteria that can do a simpler version of it. But that process doesn't split water and make oxygen.
Okay. So cyanobacteria split water and grab the carbon dioxide from atmosphere.
Yes, and then they get the electrons off of the water and they add that to the carbon dioxide, and that's how they can make carbohydrate.
Yeah, so carbohydrate or sugar.
Yes.
And then the other part of living forms is nitrogen. So some cyano can...
Yes, cyanobacteria aren't the only organisms that can do this, but we have an atmosphere that has a lot of nitrogen, but that's not nitrogen that can be used by plants or animals. It has to be in a form other than gaseous nitrogen. And there are some microorganisms that can take the nitrogen out of the air and combine electrons with it and make nitrate, and ammonia, which can then be used for amino acids and nucleic acids like DNA and RNA. These are nitrogen compounds that life really needs. And some cyanobacteria are able to pull the nitrogen out of the air and convert it to this usable form.
So that means all the fundamental things in biology, carbohydrate,
right?
Amino acids are proteins,
right?
And then nucleic acid, DNA and RNA. All of them trace back to the splitting water and fixing nitrogen.
Yes, absolutely.
And you made a comment that no plant can fix nitrogen. So that means all the nitrogen that's fixed that we have in our body that indirectly comes from plants or animals, they ultimately come from microbes.
Yes, they have to in some way or another cooperate with microorganisms, and also some is just created by lightning. So there are geophysical processes also, but for biological production, it's done by microorganisms. Yes.
Yeah. So I guess that was the key moment in modern industrial revolution in early part of last century when this nitrogen fixing reaction
Yes.
was invented.
The Haber process.
The
yes.
Haber process that essentially we could fix nitrogen with to make ammonia and then
Right.
urea. So that revolutionized, and almost I think almost all of us, if we trace back our nitrogen, partly comes from that reaction.
Oh, I'm sure. Yes, it's a very big business in the world.
Yeah.
Ever since it was developed.
But at least we can say that, can we say for sure then, or with some certainty, that the cyanobacteria are the mother of all modern life forms, or at least the...
That's a little much, because there are other bacteria that were with them way back when, and
Okay, so the members of the ancestral family.
yeah, we'll go with that. I think that for organisms that can use oxygen and tolerate oxygen, then they're the oldest.
I see. So since they were fixing the carbon dioxide and producing oxygen, were they responsible for increasing the oxygen concentration in our atmosphere?
They were entirely responsible for that. So the great oxidation event geologically was from cyanobacteria. And at that time, even though we think of oxygen as being this wonderful thing that we need, it was the single most cataclysmic event in the history of the planet for life, because it killed off almost everything on the planet. Oxygen is corrosive. We now have evolved organisms on the planet that use oxygen, that need oxygen, but that was not the situation before the cyanobacteria. And there were other organisms, microorganisms on the planet, and they could not tolerate oxygen. So there was a lot of death and destruction, a bottleneck, because of the creation of the oxygen in the atmosphere. But absolutely, cyanobacteria, they're still responsible for somewhere around a quarter to a third of the oxygen we're breathing right now. It is because of cyanobacteria in the environment.
But when you say a lot of organisms died, they were not the fish or...
No. They would have been some of the very earliest organisms on the planet had other kinds of metabolism. Yes, anything that uses oxygen was not around until well after cyanobacteria.
Oh, okay. So they essentially...
They invented the ability to tolerate oxygen, yes.
And then that oxygen, this carbon dioxide splitting, water splitting and carbohydrate making process, or ATP generating process, that starts with what is called photosystem, right? So
yes. So there are other photosynthetic bacteria, and they have one of two kinds of processes that can be used to gain energy and fix carbon. What cyanobacteria do that's special is they put these together in a chain and create a much more efficient process that uses water as a source of electrons. And we still have some of these organisms that have only one kind of, we call it a reaction center. There's some that have one that's like what plants and cyanobacteria call photosystem 1, and there's some that have a reaction center like what plants and cyanobacteria we call photosystem 2. But plants and cyanobacteria have both. It's a much more efficient process, and it really doesn't need any organic materials supplied. It can use really water and CO2 from the atmosphere. And so it's that efficient putting together of two kinds of photosynthetic complexes to make this very efficient chain. This is what was invented by the cyanobacteria.
I see. And right now, if someone is curious to visit some cyanobacteria, what are they? Where do they grow?
If we went outside right now and looked up at buildings, and you could see some kind of gray streaks where the water drips a little bit, those would be cyanobacteria. If you see lichens, those are a symbiosis of cyanobacteria and fungi. There are cyanobacteria in every body of water, in every bit of dirt that you could dig up. They're in the Arctic, they're in the Antarctic. They help to form the crusts, desert crusts that are what keep all of the dirt from the sand from blowing away. They're in every habitat on the planet. Some are in I don't think any cyanobacteria have been found in the extreme hydrothermal vents, but in Yellowstone there are in the they're in the hot pools. They're not boiling, but a little away from the boiling ones, you can find cyanobacteria.
So that means they're almost
everywhere. They're everywhere.
And
they're really everywhere.
Fixing carbon dioxide, producing oxygen.
Yes. So.
So because, as you said, nearly 1/3 or 1/4 of all the oxygen on this planet.
Yeah, right this minute.
Right this minute. So they're
coming from cyanobacteria.
They are
doing their job.
And they're making us breathe and have a normal life.
Right.
Yeah. So coming back to this question, or the point that you made, that you discovered this very important gene that is involved in this carbon pictures. So, yeah.
Actually, in the photosynthesis has two parts. One of them is the sunlight energy capture, and the other part is the fixing of carbon from the atmosphere. And so the so-called light reactions are the ones that get solar energy. And that's the special one. The water split, the part of photosynthesis that splits water and pulls electrons off of it, that's the kind of the thing that was invented by cyanobacteria. And the heart of that little machine is there's a pair of proteins that are able to let the electrons move.
Yeah.
And one of those two, actually I ended up working on both of them, but one of those two proteins that fit together like this is the protein that I was working on. And so I was very lucky to be a graduate student and a postdoc at the time that how that all works was being revealed by a number of laboratories, some of which were not working with cyanobacteria, but with some of these other kinds of bacteria that just have one type, because the homologs were identified, that there was a version in the cyanobacteria and another version in the ones that can't produce oxygen. But so I was definitely there at the right time when all that was unfolding. Before that, so I think most students who take a biology class and learn a little about photosynthesis, or they take a biochemistry class, they learn the Z scheme of photosynthesis. And that's, the path the electron follows. But I was shocked to learn when I was in graduate school that those were just signals on different kinds of scopes. Those were biophysical signals in the Z scheme. It was not known what the components were that were responsible. So the components in the membranes of plant chloroplasts and in cyanobacteria that were doing the photosynthesis, it actually wasn't known other than that you'd get a particular kind of spectroscopic blip. The Z scheme was developed without actually knowing what the components were that was doing it. And so I got to be a graduate student and postdoc while that black box was opened up. And fortunately, it was opened up and one of the proteins I was working on was in it. And so that helped me get off to a good start, because I started then working on how those genes are regulated. And that was how I got my independent start in science.
Yeah, so I guess that's the exciting era of molecular biology, because that's when, as you said, people knew that these reactions might happen, but they don't know what
How they happen, yes.
or how they happen.
Right.
Similarly, in physiology or in biochemistry, there are also many reactions that are known, but people didn't know which genes and which proteins.
Right, exactly. So,
And I think that's the time from maybe 70s till maybe late 90s. That's when people are trying, those are the era of gene discovery, when people wanted to know what are
Right.
the genes and how can we get to them. So that was the exciting.
absolutely. And, and I was very fortunate to be in the right place first for photosynthesis and then in the right place for circadian biology of, figuring out what those genes are.
And you mentioned that you try to figure out how to make transgenic cyanobacteria. And these days we are talking about genetic engineering or curing disease with changing our genetic code. But now dial back maybe 30 years, maybe 80s, right? That's when the transgenic cyanobacteria were made.
Yes, actually, I started graduate school in 1978 and got my PhD in '83. So I was in fact making cloning vectors for this organism in that period of time. I should say this is such a wonderful, the specific cyanobacterium I work with, which is called Synechococcus elongatus, is such a wonderful little creature to work with. It's so easy to transform. It takes up DNA. If you have engineered your DNA right, it will gladly put that in the chromosome where you want it to go. It has a little tiny genome, smaller than the genome of E. coli. It's very well behaved. So I got very lucky, and initially we haven't really gotten to the point of talking so much about circadian biology, but in retrospect, it was so lucky that we started with this species because its circadian properties are also so much easier to study than in some of the other species we could have been looking at. Yeah,
so this photosynthesis, how did you get on circadian rhythm? Because I imagine that photosynthesis happens when there is sunlight.
Yes. So I was studying photosynthesis genes, these genes that are involved in photosystem 2, one of the part of the photosynthetic machinery where water is split. And in this bacterium that has fewer genes than E. coli, it had 3 genes for this protein. And that was, at that time, that was really unusual to find in bacteria, that they would have what we would now call a gene family. That was, unusual, especially when it's so streamlined otherwise. Chloroplasts only have 1 gene, one copy of this gene. But my cyanobacterium had 3, and so I was focused on why and are they regulated differently, and they were. And the bottom line is that there's one that's we would call the housekeeping version. This gene's called PSBA. This was the housekeeping gene, and under most conditions it was expressed a lot, and the other 2 copies were almost off. But if you would give the cells high light, expression of the first one would go down and the other 2 would come up. And so it was looking at this regulation is what I had established my early lab based on. And the tools that we made to study that regulation turned out to be really useful for looking for the components of the clock later. And just to finish the first story, it turns out that this housekeeping version, it makes a protein that is very efficient, but can be damaged by light. And the other 2 that come on at high light are not as efficient, but they're they withstand high light better, less light damage. And so that turned out that it took a long time for that story to completely unfold, but it's the tools that we made to study that process that made it possible later to study the circadian process.
So that's very exciting. So what you're saying is cyanobacteria makes at least two different versions of this, very important, at the heart of all our energy need.
Yes.
And one is like the pilot burner in our water heater that is almost always on. But when so for example, overnight it may be on just to keep the cyano just primed, so that in the morning as the sun comes up, it will slowly detect, or it will get damaged by the rising sun. When the sunlight is too much, then it will kind of turn off, and then the cyanobacteria can turn on the other two genes which can withstand very bright sunlight. And
that's generally a good way of thinking about it. Surprisingly, the damage to this protein and the repair of that damage is the rate limiting step in photosynthesis in plants and in cyanobacteria. It's an unusually light labile protein because it gets damaged by the chemistry it's carrying out. And so, and the fact that has to be replaced throughout the day is part of how it was discovered.
I see. Okay. So then I remember you said these tools that you generated to study these proteins. What are those tools?
The tools are reporter genes that can let us tell what a gene is doing. So what we had were 3 genes, and we wanted to know how much is each gene being expressed under different conditions. And at that time, the kinds of reporters that were being used were not very dynamic. You couldn't, you could look at a gene, and you could use, and I guess I should define reporter gene first. If we're looking for the gene to be regulated, that gene is going to have regulatory signals that will be able to interpret that they should turn the gene on. And we can hook that regulatory region up to a coding region that'll make something we can measure. Because otherwise we couldn't just look at the cyanobacteria and tell if a particular gene was on or not. And so at the kinds of genes that people would measure were proteins that would be made, and they'd make a color. First of all, they'd usually make blue. We couldn't see that very well on our cyanobacteria, which are blue-green. But the other thing is they would make the protein, and the protein would hang around.
Yeah.
And so if you were looking for expression to go up, that might be okay. But if you were looking that the gene might turn off, you couldn't tell it turned off because you still had this high level around. Yeah. So this was just at the time that 2 groups were starting to work with a different kind of reporter, and this was a luciferase enzyme, light-emitting enzymes like the kind that firefly make. And there were on one hand, there was a lab that was using them in a kind of bacteria called Streptomyces, and because of that work, there was another cyanobacterial lab that was starting to develop luciferase for using in cyanobacteria. And we got these genes and were able to make our cells produce light that we could measure with a very sensitive cooled CCD camera to try to see if when we would expose our cells to high light, would these different genes we were looking at come on or turn off. And this was around the same time that Steve Kay's lab, working in Arabidopsis, was using luciferase to look at circadian regulated genes in Arabidopsis. And so
that's a plant.
The timeline, yes, thank you, on a plant. And So this timing is important because this is starting to parallel the two tracks that I ended up working on, the studying the photosynthesis genes, and then ending up looking at circadian regulation and finding the genes that make up the circadian clock. But I was in this environment of knowing about this plant work and also knowing about this bacterial work and starting to use these tools. So when I'm talking about tools, I'm talking about glowing cyanobacteria, where they glow as a function of how much a particular gene is being turned on.
Yeah, so when you're thinking about these glowing organisms, there are two things that come to mind. One is the firefly, and the other one is the red algae in ocean, Gonialox and
Right.
a few other that will
Right.
glow as the waves break.
And yes, absolutely. And there, in fact, are a lot of organisms that make these enzymes called luciferases. And interestingly, they have evolved many times throughout evolutionary time. And those different enzymes are not related to each other. They don't all have a common origin. In addition to the red algae and fireflies, there are also a lot of bacteria, both terrestrial bacteria and marine bacteria, that make luciferases. And the first ones that we were using were, in fact, the bacterial type. We now, some of our work we do with the firefly enzyme, and some we do with the bacterial enzyme. More with firefly now, just because it turns out to technically be a little easier for us, using these two types of genes so that we could get the cells to glow as a function of the gene turning on, were important first for our photosynthesis work and later for our circadian work.
Yeah, I remember when I was a grad student in Steve K's lab, Steve was using this tiny Arabidopsis weeds, not the type of weeds most people are familiar with. These are small plants. And these plants were expressing this firefly luciferase, and we would put the highly sensitive cameras to take a picture. And of course, this is not your garden variety picture. We would have an exposure time of 10 minutes or 15 minutes, and the camera would stay open for 10 or 15 minutes, and the image would come in. And I remember the first time when I took the first image, although it was already 10 years of history at that time, I was super excited to see that the plant, there's a glowing plant. Of course, it is not glowing as bright as firefly that I could see with my naked eye, but when the gene one was on, it would just brighten up, and it made the work so much easier.
Oh, absolutely.
Yeah.
Absolutely.
So that was a really exciting time in biology in general at that time.
I mean, this was really the beginning of the automation of being able to study temporal things that are changing over time, and particularly if you needed to be able to find mutants that didn't do a process correctly, where you have to look at thousands and thousands of individuals that vary, and you have to look at them for a week, many times a day. The automation is critical, and this is the light production of luciferases and being able to harness that for getting these kind of dynamic readings was really critical.
Yeah, so at that time, of course, it was very less automation in the lab when I was working.
Yes, absolutely. A lot harder.
Yeah, so I remember that I was doing the graveyard shift. So that means every evening I would come in around, say, 10 o'clock and stay in the lab till 4 a.m. And in every 30 minutes, I would put on this infrared helmet. Those were Canadian Army surplus infrared helmet. And it was really cool to put on this infrared helmet, and in complete darkness, I could see things, to take these plants, put them under the camera, then shut the door tight, come out and turn on the camera, and then after 25 minutes come back and turn off the process. So it actually trained me to sleep only for 20 minutes and then come back and change the plates. So at that time, I remember that other labs were working on making this process automated, and that's the time when the scientist Takao Kondo, Professor late Takao Kondo, came up with this rotating base, and we used to call it Kondotron. And can you tell us a little bit about Kondotron?
Well, so I need to back up a little bit, and how we really ended up making the shift from studying photosynthesis to studying to studying circadian biology, and without trying to back up too much. At the time that I was approached about doing a circadian experiment, the scene had been set. My first PhD student was a guy named Mike Schaefer, who now is actually very high up at allergy and infectious diseases at NIH. But he was a good friend of Vinnie Cassone's. So Vinnie being a circadian bird expert, who was in my department at Texas A&M at the time. And they would talk a lot. And Michael already knew from talking to Vinnie about these, some reports of cyanobacteria showing circadian rhythms. And in fact, from some photosynthesis work that Michael Schaefer was doing, where we would have bioreactors that we were running for multiple days, looking at one of these clunky reporter genes, he was seeing some oscillations. But at the time I said, that's not what we work on, and this is weird stuff, and you have to really know what you're doing, and we're not getting into that. So I was not completely unaware of the circadian field, and I even knew that there had been reports of circadian rhythms in cyanobacteria. But that was still not something, it wasn't something I was thinking of focusing on. But when Takao Kondo was doing a sabbatical in the lab of Woody Hastings and Carl Johnson, and part with Woody Hastings and part with Carl Johnson.
Which university?
Woody was at Harvard and Carl at Vanderbilt. Carl and Takao were both working on plants. Carl was working on Chlamydomonas, a green alga, and Takao was working on duckweed, limna.
Really?
And Takao was he was an amazing scientist, but he was also an amazing engineer, and he would devise these various machines to try to automate measurements. And with duckweed, he was using fluorescence signals, and because he couldn't, there was no transformation, there were no reporter genes in the duckweed, and Carl was also struggling with trying to get a genetic system for Chlamydomonas, which took many more years before that all got worked out, and so they discussed the fact that instead of they wanted a simpler photosynthetic system. And they also knew about these reports of rhythms in cyanobacteria, but those were in cyanobacteria that nobody was able to transform. And so Carl had the idea of taking a different tack, which was, well, if some cyanobacteria have circadian rhythms, probably they all have circadian rhythms. And why don't we try one that is easy to work with genetically? And so Carl contacted me, and because I was the one who had the genetic system. And I said, well, we made luciferase reporters to, a highly expressed gene. I'll send it to you and let's see what happens. And so I sent it there. And while Takao was still in Carl's lab, they did some measurements, and this was back in the day of the fax machine, faxed a copy of what they saw, and it was like if you held it like this and squinted at it, you could kind of see, something. Yeah, maybe. But Takao took the strains back to Japan, and he's this master of measurement and devising machines. And the next fax I got, you did not have to squint at. It was like, it was unbelievably beautiful. And as it turns out, the rhythms of gene expression from this organism are phenomenal. They are robust, they are high amplitude, so the trough to the peak is high, they're stable, they are great.
Which year was this?
Probably 1992. I think our first paper was in 90. I think that the paper came out in 93.
Yeah.
And so that began the three of us working in earnest, and then later Masahiro Ishiura, who worked with Takao Kondo, with Kondo-san, the four of us then worked internationally to try then to have, we have these beautiful rhythms, you can transform the organism. That means we should be able to get mutants that have disrupted circadian rhythms and complement the mutants, add back genes, and find out what the genes are that are broken in the mutants. And that should be the clock.
By that time, when you were talking about 90s, by that time the Drosophila circadian clock gene PAR was already cloned?
Right. Yes. The period gene PER was already cloned, and this was before genome sequences.
Yeah.
But so we couldn't just look and see if we had a PER gene. But many people had tried to look using homology, using the methods at the time to take a labeled period gene and see if you could find it in cyanobacteria, and it wasn't there because it isn't there.
Yeah.
So there was no hint at what might make up the clock, but we were pretty sure it was going to be fundamentally different.
So you got super excited about clock and kind of wind down your photosystem.
Well, at first I thought the clock thing would be this little side project, but and for a few years I continued to believe that. But we would there were a couple of mutants we had, one in particular that turned out to be important called KIK-A, that had a little tiny phenotype with what we were looking at for it. The screen had been for these light-regulated photosynthesis genes. It had a little phenotype, and then it had this really big circadian phenotype. So at some point, it was very clear that there was a lot to be learned studying the clock. And at that time, the black box was open in photosynthesis, and the big discoveries were, it's not that there was nothing left to do. There are still questions I didn't answer from my earlier work that I would love to know the answer to. But it was not as exciting, not fundamental steps forward. And there was the opportunity to have fundamental steps forward with finding out, how does this clock work? What is this clock? And again, not that many genes, about 2,700 genes in the organism. So not that many puzzle pieces to find the right ones.
Yeah. And before we move on to clock, one last question about photosystem. We always talk about splitting water. Do we know how water is split?
We do.
And can any engineer replicate that system without photosystem? Can we artificially split water and use that to make energy, just the way cyanobacteria? There
are, in fact, groups, particularly I'm thinking of a group in Sweden that, synthetic leaf groups that are trying to do this. The short version is that there's a capacitor. So in photosynthesis, there is, when a photon hits, there's one electron that gets boosted, but you have to have 4 of those boosted to split water. It's very hard to get the oxygen off of water.
Yeah.
But there is a cluster of manganese ions that are associated with the complex that can act as a capacitor. And so it can be quadruply charged, and then bang, that's enough to put 4 electrons on. Yeah.
So that means we can one day replace plant leaf.
Yes, I have to confess I haven't really kept up with the progress, but there are definitely groups working on that.
Yeah.
Yes, there are groups doing that. Yes,
Because I always thought that these two fundamental life processes, splitting water and fixing nitrogen. Nitrogen fixation.
Yes, and that's the nitrogen fixation. There are also groups working hard on that, both biologically trying to get it into plants, and then other ways of semi-synthetically, bioengineering biological principles into synthetics. This kind of work is being done. Yes.
Yeah. Okay, now back to clock again, because there are so many exciting stuff that came out of cyanobacteria. And I think that's the time around 94 or 93 or 94, that's when you, all four of you,
Yes.
a long time,
Yes.
all four of you, or at least three of you, Carl Johnson and Takao Kondo, were publishing this very high impact, very fundamental papers for a while.
Yes, plan worked. As you had said, so Kondo-san was, he did build this Kondotron, this machine. What it allowed us to do was to mutagenize, take our reporter strains so that we were measuring the circadian expression of a gene by light production, mutagenize them and get colonies where all the cells in that colony might carry a mutation, and some of them would have a mutation in a gene that makes it part of the clock, and that would change this bioluminescence production so that it would, instead of maybe a 24-hour period, have a 22-hour period or a 28-hour period, or they'd be arrhythmic. And in fact, we found all of those mutants. We found short period mutants, lots of long period mutants, some arrhythmic mutants, and then made a library of, and this was really, Masahiro Ishiura was very into making libraries, and he made a really good one that was very easy to use. That was the one that got us the clock genes. But a library then that could be and a library being a collection of cloned pieces of DNA. So from the normal copies, the wild type copies of the gene, and we could move in these pieces of DNA and fix the clock so that if it was a 28-hour period mutant, it would go back to 24 hours, which is what you want a circadian rhythm to be. And it turned out that we had, I don't remember how many, but about maybe 24 mutants. They were all fixed by the same piece of DNA. And that piece of DNA had 3 genes on it that we then sequenced, found out where all the mutations were, but also just sequencing it, found out that they were genes that were at that time not known elsewhere in the databases. The databases were very small at that time, so there were not that many genes that had been sequenced. So they were novel genes.
But until now, also, the cyanobacteria clock is very unique.
Yes, it is very unique. It is so there are the animals seem to use variations on one kind of clock. Fungi use the same blueprint but have different components. Plants, again, blueprint's generally the same. The cyanobacteria is different. It really is a machine. It's a discrete nano machine, and this is why you're interviewing me. Is that it turned out that the circadian clock is so spectacular, and this is really jumping ahead many years now. But what makes it especially spectacular is that in 2005, Takao Kondo's lab showed that you could take the components encoded by these 3 genes, so these proteins called Chi-A, Chi-B, and Chi-C, and you could put them in a test tube with ATP, and you could get a 24-hour rhythm of the phosphorylation of one of those components, Chi-C. that it would phosphorylate and dephosphorylate and phosphorylate and dephosphorylate and just keep going for days and days, as long as anybody would care to look at it, which was at that time tedious. We have an easier way of doing it now. But everything you need to set up 24-hour time is on those 3 proteins.
Plus ATP.
Plus ATP, yes. And yes, ATP is very important. And that's because one of the components, Chi-C, which is at the heart of all of this, has an ATPase activity. So it hydrolyzes the ATP super, super slowly. And that's actually part of the timing system. That's a critical part of the timing system, actually.
But I want to kind of wind back.
Yes.
The clock.
Yes, wind back to clock.
To 90s, and then that's when, again, this consortium kind of published one very provocative paper that almost every gene in cyanobacteria was going up and down.
Yes. So that was actually that was quite fun paper. The way that project was done was by taking this luciferase gene, but instead of hooking it up to the regulatory region of some particular gene we were studying, using a transposon, which is a jumping gene, and letting that transposon carry this luciferase gene into the cyanobacteria and just randomly put it in somewhere. And because this luciferase gene didn't have its own regulatory region or promoter, it couldn't turn on unless it jumped into a gene and hijacked its regulatory region, and we got thousands and thousands and thousands of these resulting glowing colonies where the luciferase was telling us what each regulatory region in the genome was doing. And every one of the colonies that glowed, it glowed with a circadian rhythm. So we have never seen, at least it's very sensitive when you're using the luciferase. It's quite sensitive, but we have never seen a gene that is not expressed rhythmically. And most of the work that's done in eukaryotes, in mammals, plants, fungi, most of that work is done by extracting RNA and doing measurements that look at the level of the RNA, which is less sensitive, and usually you can't get as many time points as we can get. So I think that the reason that I would say 100%. At the level of transcription, it's 100%.
Yeah.
And in papers, there'll be, at least 80% or something, and I'm going, it's all of them. It's just all of them.
No, I remember that paper, and then that also is a testament to 3 or 4 very fundamental things in biological research. One is the technology expands our imagination, and we can ask questions that we could not ask before.
Absolutely.
Just like you said, all these other ways to figure out whether a gene is cycling is manually taking the plant or the tissue, grinding up and doing something. And you can, if you have a great team, then maybe you can do it once an hour or so, and maybe for a couple of cycles. Mm-hmm. But with the Kondotron, you could actually put hundreds, if not thousands, of colonies,
yes,
and then walk away and come back after a week.
Yep, exactly. So the Kondotron was a disc that had, I think, a dozen places for petri dishes, and then each petri dish could have hundreds of colonies on it, and this disc would turn to position the colony under the cold CCD camera, which could measure the light production. And if a plate wasn't being measured, otherwise it was in constant light, because we didn't want a light-dark cycle. But we were measuring light production, so it had to, for a few minutes, be masked off and go into the dark so the machine could measure how much light are the colonies giving off. But because it would rotate, it could reposition that plate every 45 minutes or so underneath the camera again and position it exactly so that for every one of those hundreds of colonies, we would get a absolute light measurement off of it for days.
Yeah.
Like you said, set it up and leave and see that the light production is going up and down with a 24-hour rhythm.
Yeah, I think that experiment has not been replicated in any other organism, not even in flies, that no one has put randomly a reporter gene in front of every promoter or regulator region and then checked whether that gene cycles or not. So I guess this is from the 90s, but it still stands the test of time.
Yes, yes, it does. And it was in kind of old school genetics, because the idea of using a transposon instead of going in and making the gene yourself in the lab, how most things are done these days. But, when you can let genetics do the work for you, it's so much more elegant than going in and seeing what you can make. Because if you set up your screen or your selection appropriately, nature tells you what you have.
I think that time I was a PhD student in scripts, and then you were giving a guest lecture on Wednesday morning. Usually Monday and Wednesday we have 2 lectures, and Monday is the scripts faculty who teach us, and Wednesday is guest lecture. And you came and gave that lecture, and at that time almost everything that we are dreaming of doing in plants or animals or mice or Drosophila, you were already doing that. So I clearly remember you made a point that if you can think of a genetic experiment in cyano, then you can do it.
Yes.
Yes. That was a very powerful statement, and I really envied you. At that time, I was thinking, wow, the cyano people are so ahead of our time. And then another thing that happened, that when you were giving that lecture, I was in the front row, and I was thinking myself, look, there is a difference, there is a distance of only 10 feet between you and I. And I was a grad student, and you are a very successful faculty. At that time, you're already a professor at Texas A&M and had really impactful papers. So then I was thinking, well, to cross this 10 feet, to stand on that stage one day will take me at least 10 years or so from then on. And fortunately, it happened in 8 years. But then that actually gave me that inspiration. Like, here is somebody who is making all this cool. If you can dream of the experiment, you can do it. So that was very inspiring, I must admit. And I guess going back to all genes are circadian, few years later, there was a review article where you were co-author and Deb Bell-Pedersen is first author, I think. We're talking about how in cyanobacteria, since it has to do everything within a single cell, then it makes sense to connect all genes to circadian clock, because the cyano has to really go to sleep and wake up in the next morning and do that stuff. And as multicellular organisms evolved, they had separation of function in different organs. You don't have to actually drive all the genes to turn on and off in every single cell, but just turn on and off the necessary key rate-limiting enzymes or rate-limiting genes in a specific tissue. So in that way, if you combine which genes are cycling in tissue A, B, C, and if you add up, then it will also come to nearly 100%.
Yeah, that's a very good point. Yeah. So some of your work looking at what's cycling in which tissues. Yes, that's a very good point. There may be some genes that aren't cycling here, but they are cycling over.
Yeah. So I think when you published that paper in 90s, I have that in mind. So when initially when we did the transcriptome studies, and then we said, oh, well, only 10% of the genes were cycling in one tissue, and people would make those bold claims that, look, cyano controls 100% of the genes, but then not a mouse liver. And then I used to say, no, wait a minute, we have only looked at mouse liver. Why should the liver care about which gene in the brain, which should cycle in the brain, should cycle in the liver. So we should look at the brain, and slowly, I think we have come to a consensus now that yes, just like in cyano, maybe in humans also. I think Felix Neff published a paper a couple of years ago showing that nearly 70-75% of genes, if you look at different tissues in humans, may be cycling. And as there are at least 200 different cell types in humans, it may be close to what you predicted almost 30 years ago.
Right. But the other thing that you alluded to earlier is how important the technology is. And the other thing is that that having technology that can take the samples very close together, the ability to detect a rhythm, it matters so much how often you sample, whether or not you can detect that the rhythm is there. And we also had that advantage that for a lot of the things that we've done, we were able to do this automated, rapid sampling that's not possible if you have to extract all the RNA out of a tissue and make cDNA and do all of the steps that you need to make there. Yeah.
And then the other thing that came from cyano, or maybe you can tell us, what are the other fundamental principles of circadian rhythm that have come from cyano, or at least cyano contributed a big way?
We've been talking a lot about the genes and discovering that there is a clock and what it's made of. I've barely mentioned the fact that we can have a clock in a test tube now, and so now we can automate that thanks to the work of Andy Lee Wong's lab. So Andy Lee Wong and Carrie Parch and our group worked together to be able to get automated information in high throughput coming out of these little 3-protein clocks, and then expanding them to be 6-protein clocks. This has really let us understand some principles that actually even go beyond circadian biology. The slow biochemistry, the fact that the circadian clock takes 24 hours, when I first got into the field and just thinking about how everyone viewed clocks, I think the assumption was that you'd have pretty ordinary processes, but you have such a relay of them that it can take 24 hours to complete the relay. The cyanobacterial clock, where it's just these 3 proteins, and we know in exquisite detail now what's happening as the hands of that clock turn, the gears of the clock turn, I should say. There is some real slow biochemistry, and where you have reactions where the 2 players are just a little bit too far apart until you bind something, and that makes these things get a little bit closer together. So I think slow biochemistry people are now finding elsewhere. And particularly this protein Chi-C that's at the heart, even though it was a pioneer protein, it is, meaning when we found it, there wasn't anything like it. Now there are genome sequences everywhere. We can tell that there are some things like it, and there are some other things like it that are not Chi-C, but that they may also have this very slow biochemistry. So this goes even beyond circadian biology, that there is slow biochemistry that this system helped to work out.
So let's kind of unwind and then maybe unpack it. So the ultimate goal of all biologists studying any process is to recreate that process in a tube. So just like when you're thinking about most biological processes, whether it's an enzyme that breaks down, say, protein into amino acid, or an enzyme that breaks down glycogen to glucose, we essentially take that protein, purify it, add some glycogen, and make sure that the glucose comes out. Then we say, aha, this is the mechanism how glycogen is broken down to glucose. So similarly, I think the holy grail of all biology is to recreate the biochemical or the process in a tube in the absence of cell, and you have a very predefined cocktail, you can say. And in that context, almost all biology until now, all biochemistry until now, is to focus on enzymes that actually do these things much faster. You can you can put glycogen on a tabletop, and maybe slowly, due to decay, automatic decay, maybe it will take days or years to break down that glycogen to glucose. But then the enzymes make this process so fast that you can see it within few seconds or even minutes. But a circadian clock, I remember, actually I remember a cartoon that's in, or a picture that is in this book, beautiful book called Time, Love and Memory, when in 1970 or 71 in a Cold Spring Harbor Symposium, someone is telling when period mutants are identified, a Nobel laureate, I'll not name who that is, says, I don't believe that the circadian clocks have a genetic basis. These are mostly driven by sun and the moon. And not only to get the genes, but to recreate that clock in a tube. And I remember in 2005, I think I had just started in Salk, and I had this office downstairs, and the paper from Takao Kondo came out, where he mixes these 3 different proteins and ATP, and there is a nice rhythm. And why this was so profound, so fundamental, was people have shown, for example, how glycogen enzyme may be breaking down glycogen to glucose, but there is no system in test tube, that actually does the reverse in a continuous fashion. So I think in that way, this was a fundamental milestone that you can really define. These are the parts of the clock, because if you take these parts, put them together, then you can make a clock.
They are sufficient, yes.
They're sufficient. They're necessary and sufficient.
Yes, absolutely.
And so that's why this is, sometimes I feel like, okay, so this should have been given a Nobel Prize, because for the first time, anyone had shown 3 different proteins making this loop, not reaction going only one way, but the loop that is repeating itself over many times. And this is, so far, I think, as you said, is the only system where this has been shown.
It's still just kind of mind-blowing and spectacular. And once he demonstrated that that could be done, it allowed so much work to be done on the mechanism of, well, how are they telling time? And this has really been, that then helped lead to another collaboration that I've been involved in now for many years, of figuring out really what's going on in that test tube. And that's been our work with Carrie Parch and Andy Liu Wang. Especially, we've been working now with Andy for 25 years or so. And really figuring out what are the, what's the progression of the gears. And this has all been made possible by the fact that you could reconstitute in a test tube. And so back to what some of the fundamental things are that we found. One of the things also that Andy discovered is that one of the proteins, the smallest one, Chi-B, which was also the most mysterious, we knew what its effect was, but we didn't know how it was doing it. Turns out that Chi-B, which had been, we knew the structure, it had been crystallized by several labs. We had NMR solution structures from different labs. We thought we knew what the tetrameric structure of that protein was. Turns out that was the inactive ground state, which is how it spends most of its time, laying around doing nothing. At a very low frequency, instead of a tetramer of 4 subunits, identical subunits, it goes down to 1, which unfolds and refolds into a completely different 3-dimensional shape. So it's a metamorphic protein, which means it's a shapeshifter. It's a fold switcher. We call it fold switching, and this is happening at a very low frequency all the time, and it just immediately goes back to that inert ground state, unless the other gears of the clock have progressed to a point that phosphorylation that Takao Kondo showed in a test tube on Kai-C, unless it's gotten to a point that has changed the shape of Kai-C enough to make a binding site, a landing pad for that fold switch protein, where it'll grab on. And then it doesn't just go back. And this fold switching is also, I started to say very unusual. Reports of a fold switch were very unusual before this protein. And now there is a little cottage industry of trying to see how to go into the databases and predict that there are metamorphic proteins there, because they are probably a lot more frequent than have been recognized, which means that you have these completely different alternate structures, and where spending more time in one structure than the other can lead to a time delay.
So you're figuring out how these 24 hours clocks are built, because in most biological systems we go from neurons firing at 5 Hz, 500 times they're changing, they're repeating the same thing, to cells dividing in 24 hours, but that's a different type of mechanism, and nobody has reconstituted cell division like this. But this is really, the detail is as complicated as a regular clock, human-made clock that does not work, like not a digital clock, but actually analog clock. A lot of things have to work in precise detail to produce this 24 hours.
You'd be surprised how many little steps there are, and we already know about them.
Yeah.
It's hard to even describe what all of the steps are. And all three of the proteins have alternative structures. They're not all fold switching like Kai-D, but there's so many little steps, the little teeth of the gears that are turning, and each step makes the next step more energetically favorable and the previous step less energetically favorable, so that you keep the clock going clockwise and not counterclockwise.
Yeah.
So all of this rather extraordinary degree of biophysical understanding is a direct result of Takao Kondo reconstituting, taking this bold, this bold step of putting these three proteins in a tube. And I have to say that, that required a lot of iterations. There was a lot of effort that went in from his laboratory to try getting the concentrations just right and getting it so that would work. But it's been extraordinary.
But that's a fundamental thing, I guess, would be unique to cyano clock, because the other clocks go back and forth between nucleus and...
Right, yes, very different.
But still, it is extremely important that this kind of mechanism predated the modern clocks by at least 1 billion years.
Yeah.
Let's put it that way.
Yeah.
Then the other fundamental principles as it relates to eukaryotes, and just like we started with, all genes are circadian. Similarly, this cyanobacteria, if we dial back 3 billion years, then there was no land. They're just floating around in ocean and maybe exposed to sunlight. They still have DNA, and there is some idea that clocks evolved as a flight from light, because light can damage DNA. Do you still believe in that concept?
I think that for a lot of organisms that makes a lot of sense. In the case of the cyanobacteria, that doesn't seem to be what they do. It doesn't. They're really good at protecting their DNA from UV damage, which is maybe not surprising because they make their livelihood in the light. They are really good at processes of repair, And they do, in fact, replicate their DNA in the daytime when light and energy are abundant. They just also have to be really good at repair. So I think that for cyanobacteria, and I say cyanobacteria, but we are talking about a tremendously diverse range of organisms, some of which are multicellular, some of which are single-celled, and that in a range of processes that they can carry out. So there's a lot of diversity there, and we really only know a lot about one of them, but from the circadian point of view. But in general, cyanobacteria are they're kind of hard to mutate with UV light. They're very robust, and so.
So they divide in nature. Do they divide their DNA during daytime?
They do.
They do.
They do. Okay. So in fact, our species doesn't divide at night, and how frequently it divides depends on how much light it's getting during the day. It will divide more frequently if it has more light. This reminds me of one of the other you were asking some fundamental lessons that came from it. Others had looked for circadian rhythms in cyanobacteria and not found them before. A lot of people didn't think it was worth looking in bacteria for circadian rhythms because they can divide more than once a day. And there was this general thought that if your life cycle could be completed in less than a day, why would you need to keep track of a day? That would be like saying, well, you can keep track of 500 years. Well, you're not going to live 500 years, so why do that? But this was another part of the work that came out of the combined Japanese Vanderbilt Golden Laboratories, was showing that their circadian rhythms keep 24-hour time even when the cells are dividing every 5 or 6 hours. So the great-grandchildren know what time their great-grandmother thought it was. They keep this timestamp really well. So they can divide rapidly when they're given abundant light, and they know 24 hours. And so that means that we don't know how the cell biology of how this is working, but when they divide, they know exactly what time it is, and they keep that timestamp even when they're dividing. So the premise was wrong, that you have to have a lifespan more than a day to care about a day. It's just not true.
Yeah, but then since these proteins are, and this, actually there is no nucleus. So.
No nucleus, yeah.
So they can keep track of time.
Yes.
When the cells are still dividing.
Yes.
But when, so there is another interesting experiment from cyano. I guess it was done first in cyano, when you took cyanobacteria that had a 22-hour clock and 24-hour clock, and if you mix them up and grow under 22 hours a day, night cycle, then...
Yes, so I'm really glad you mentioned this, because I do think that this has been one of the great contributions from the cyanobacterial system, is that from the beginning of the time that people started looking at circadian rhythms in organisms, there has been this expectation that, theoretically, and it should be true, that having this circadian clock gives a fitness advantage. But it had not been demonstrated. And Carl Johnson had this wonderful idea, because we had all of these different mutants, and because you can mix, because they grow in a culture that's a population, that we should be able to mix mutants that have wild type and mutants that have different circadian periods, and ask, does it matter? And that you can start with a population that's 50-50, and then you can grow it in a day that's 24 hours or 30 hours or 22 hours, and ask who wins. And it turns out that wild type doesn't always win. If a mutant, if you are growing in a 30-hour day, that would be a light-dark cycle to simulate 30 hours. A 30-hour period mutant will beat wild type. And and the same if you go with a short day and you have a 22-hour period mutant. And it's really striking if you take the two mutants, they can, whichever one matches the light cycle you're giving it will, almost completely beat out the others in the population. And this demonstration of what people expected to be true, I mean, this was a validation of the whole field that circadian biology matters, that when your internal clock matches the external day, you're better off.
Mm-hmm. So let me ask you a related question. Suppose say you take 22 hours clock.
Mm-hmm.
So that means it will win best if there is 11 hours light, 11 hours darkness.
Mm-hmm.
What if you grow it in 11 hours light and 13 hours darkness? Or 13 hours light and 11 hours darkness? Will it win over a 24 hours clock?
Photoperiod is a different question.
No, the reason why I'm asking is, in humans, most of us have roughly 24 hours, mm-hmm. 15 minutes, or 25 hours. And even if we find a mutant human, mm-hmm. which has a 22 hours clock, for example, we cannot find a new planet with 22 hours light cycle. Uh-huh. But the question is, will that person do better?
I don't think so, and I'll tell you why I don't think so. My own idea, my own thinking about why it makes sense that circadian clocks are only circadian, only about a day, is that you need the selective pressure for being able to reset the clock. And that's because of what you just hit on, which is photoperiod. That other than being at the equator during the seasons, there's a change in when light comes and light leaves. And whatever the behavior is that the organism has needs to change. I actually think this is why it makes sense to have, well, it's not the only reason, but one reason it makes sense for even a fast dividing organism to have a circadian clock. There's been some other lovely work that came out of Carl Johnson's lab recently showing that by giving a winter photoperiod, you can make the cells be more cold hardy.
Yeah.
So I think that having this ability to track dusk and dawn, even as the photoperiod changes, is important. And if you had, I don't know, with a given human, it might be different, but particularly when you can look at big populations, I think that if you had an organism that that had exactly a 24-hour clock and it didn't ever need to reset and it was always in these great perfect conditions, as soon as those conditions change, it would not be fit anymore, and it would perhaps completely lose the ability to make any adjustment. And you were asking earlier about nitrogen fixation. Just one of the things that goes through my mind is that if you grow cyanobacteria in the lab on fixed nitrogen, give them ammonia or nitrate, cyanobacteria that can fix nitrogen, pretty quickly your population gets taken over by things that can't fix nitrogen anymore. If they never have to do it, those genes are too expensive. Mm-hmm. And I think that having the resetting mechanism be selected for every day ensures that it works. Mm-hmm. And then you have it when you need it.
I see. So since you brought up nitrogen.
Yes.
And since you cannot, because of the biochemistry of it, nitrogen fixation is, it doesn't need oxygen. In fact, oxygen is toxic to nitrogen fixation, whereas splitting water produces oxygen. So then cyanobacteria that can fix nitrogen, how do they do it?
Okay, so this is actually how circadian rhythms were found in cyanobacteria. It wasn't from anybody looking for circadian rhythms, and it wasn't me, which I sometimes get credit for that. I did not find it. But what had been known for a long time is that there are filamentous strains that have many cells in a chain. So a lot of a lot of people have looked under the microscope and they've seen these beads on a string, Anabina. This is a cyanobacterium that is multicellular in a chain, and they can fix nitrogen. And if they don't have fixed nitrogen, what happens is that about every 10th cell differentiates into a different cell type. These that are called heterocysts. These heterocysts kind of seal themselves off. They seal themselves off a lot from the other cells. They don't undergo oxygen-producing photosynthesis anymore, but they turn on the nitrogen fixation genes. And then the vegetative cells that don't do this differentiation, they do photosynthesis that gives off oxygen, but they don't fix nitrogen. And then the cells cooperate. So the fixed nitrogen is sent to the vegetative cells. The vegetative cells send fixed carbon to the heterocysts. That's fine if you can differentiate and separate, but it was also known that there are unicellular strains that can fix nitrogen. What do? They can't wall themselves off. They have to make oxygen. And as you said, these are incompatible processes. So the nitrogenase enzyme is notoriously oxygen sensitive. So it requires an anaerobic or nearly anaerobic environment. So it turned out that the way unicellular cyanobacteria that fix nitrogen do it is that they undergo photosynthesis during the day when the sun is out, and then they only fix nitrogen at night. And if you have them in a light-dark cycle, you can see this very strongly, but if you keep them in continuous light, they keep alternating those. So they'll turn off photosynthesis and turn on nitrogen fixation.
Even in continuous light.
Even in continuous light, yeah. So that discovery was, in fact, what made it clear that cyanobacteria have circadian rhythms, was the fact that they would do this oscillation even when they weren't in a light-dark cycle, and that's how a unicellular strain separates these incompatible processes. It can't separate them spatially, so it has to separate them temporally. One of the ironies here is that Synechococcus elongatus does not fix nitrogen. So here we have this great origin story, but the model system that let us understand the clock is not the one where this origin story process was in fact discovered.
I see. So now you made one comment at the beginning of this discussion that cyanobacteria, the ancient cyanobacteria, when it started splitting water, produced oxygen. Oxygen is very corrosive, makes very life miserable because it can oxidize many things.
I think rust.
Rust. And
it rusted the planet.
It rusted the planet, but also inside our cells, when there is too much oxygen radicals, then that also damages proteins, damages DNA and many things. And that's why we are always driven by this curiosity to swallow some antioxidant to calm our oxidative processes. So what you are saying is having a strong circadian rhythm, at least for this unicellular cyanobacteria that fix nitrogen, is a nature's solution to have an antioxidant.
Yes, that's true. But even when you don't have the incompatible processes, what we've been able to discover in the more biological end of my lab, rather than the more biophysical and biochemical part, is that what the clock does for it is it separates out the cells in the morning. First of all, get ready to deal with all the light that they're going to get. As they're photosynthesizing during the day, they are packing away glycogen, which you know a lot about glycogen. They're packing away glycogen, they're storing that. But also I mentioned that the photosynthetic process is very hard on the cells. So there is oxidative damage that's done to the photosynthetic machinery. A lot of these reactive oxygen species that are corrosive are made. And so at the time the cells get ready, the sun sets and the cells get ready to go into the dark, there's a lot of this ROS, reactive oxygen species, around. And then when the lights go off, they don't have the ATP that they need. Well, actually, they don't need reductant that they need to mop it up. So what they do is the clock turns on genes that code for enzymes that break down glycogen, and they use the breakdown of glycogen to make the reductant that mops up the ROS.
Oh.
And if we have cells that can't do that, they are dead and within 3 hours in the dark.
Wow.
So in the day, they're fine because the photosynthesis makes reductant. They're accumulating the damage, but they also are making the reductant, and they're mopping it up. But if they go in the dark and they cannot turn on these genes that are critical for mobilizing glycogen at night, ROS kills them within a few hours.
Wow. I should actually go back to some of the mouse experiment we did, because in time-restricted feeding, which is now popular as intermittent fasting, we do see the mouse liver accumulates a lot of glycogen, because in electron microscopy, you can see the glycogens light up if you have the right high-resolution electron microscope. And then, of course, the glycogen is used up when the mice go through their over-day fasting, because mice are nocturnal. They fast during the daytime. And we know that there is less oxidative stress in this mouse liver. So maybe the glycogen breakdown, I got to go back and look up the biochemistry textbook and cyanobacteria, how it does.
Oxidative pentose phosphate pathway.
Okay, so oxidative, yes.
I can send you a nice slide.
Actually, the pentose phosphate pathway, the flux to the pentose phosphate pathway actually goes up under time-restricted eating. So that's what we had seen a long time ago in 2012 in our first paper. We had seen that. So it will be good to go back and link all of that, because we did see the flux through PPP went up. So this is one interesting mechanism, that the clock temporarily separates incompatible processes. And second, as part of that, it also produces the mechanism that mops up or cleans up many oxidative damage causing reactive oxygen species.
Right, and just like what you see with mammalian metabolism, in addition to separating incompatible processes, you just need an appropriate progression of processes. And so at the end of this point where all of this ROS has been accumulated, you need the ability to mop it up. And so having a progression of processes, each of which either fixes something that was damaged before or sets the stage for what comes next, is it's not surprising that having a clock that matches the environment is important.
Yeah. So these are pretty exciting. And then recently I saw your very futuristic, means going from cyano, which have been here for 3.5 billion years, into the human frontier now is to go colonize another planet, or at least spend few days or maybe months in space travel. And then the question is, how can you have a good space companion that can produce oxygen and maybe split the water, make carbohydrate and fix nitrogen, so what's new?
Well, so cyanobacteria are definitely being considered by several groups that are funded by NASA and other international agencies as a photosynthetic, small format, not soil requiring, organisms that you could use to produce food. And like you said, fix nitrogen, use wastewater. So I'll regenerate atmosphere, suck up CO2, produce oxygen. It's kind of everything that you would need for regeneration systems on another planet or in a spaceship. And so I think the paper you're referring to is one that we did in collaboration with my husband's lab.
Yes.
So Brian Bichet, who was a postdoc with Jim at the time, and has continued doing some circadian work with me. He was really interested in this space idea and the fact that the shuttle actually has a very fast light dark cycle because it goes around the planet
90 minutes.
Yeah, every 90 minutes. Yeah. So it's 45, light dark. And so he had a question of whether, would the clock be good or bad, and what genes are important for being able to survive in this 90-minute day. And he used this wonderful tool we have, which is we have this population of mutants that where each mutation is barcoded with a particular DNA sequence, and so he was able to simulate being on the space shuttle, having this 90-minute day, and seeing which mutants fared better and which fared worse. And there weren't any really that were particularly better, but the ones that were worse were in glycogen utilization. And interestingly, it wasn't an ROS issue. So it really turns out to be different. But they needed to be able to store and utilize glycogen to be able to deal with this very short day.
So that means there is still hope that you can re-engineer cyano, and you can stick that cyano in a culture flask, I would suspect, and it would be stuck to one of the windows of the internal station.
Yeah, absolutely. I think it's figuring out how to scale everything, and but they're much more promising than most organisms for engineering methods for trying to deal with all of this regeneration that you need to deal with. And of course, but the idea ultimately would be terraforming Mars. I mean, they terraformed Terra. I mean, they're the originators of our atmosphere, and so could they do it again? Absolutely.
Yeah. So is it possible that the cyanos are from Mars? I mean, from other planets?
It's certainly possible that precursors are from other planets. I rather suspect, because of our kind of more or less unique oxygen atmosphere, that maybe they were invented here, but maybe from organisms that came from elsewhere.
Yeah. So while you have been extremely successful in science, you also came to UCSD when almost 20 years ago?
16, yeah.
16 years
Getting
ago.
close, yeah.
And I think before coming to UCSD in Texas A&M, you had started a circadian biology, chronobiology center.
Yes, I can't take credit for that. I would say that we have had a really strong group, and actually that group continues. So Paul Harden, Deborah Bell Peterson, at that time Vinny Cassone was still there when we were there. So there's been a strong cohort of circadian biologists there for quite a while, and that center is still going strong.
Yeah, because I remember when I was applying to circadian rhythm centers, I had also applied to Texas A&M. Mm-hmm. And Vinny Cassone, till date, he always tells me, well, if he was a little bit quick enough to accept me, because University of Virginia sent me the acceptance letter, and I was hungry because I wanted to move down.
We've all been kicking ourselves, Satchin. We all look back on that and say we should have rolled out a red carpet for this guy.
No. So the point is, you actually Texas A&M. I think at that time they had a circadian rhythm undergraduate program. Did they have a class?
I don't think a graduate class, but not an undergraduate class at that point.
So that means when you came to UC San Diego and you started this undergraduate class.
Actually, Stu Brody and Michael Gorman started the class.
Oh.
And when I got here, Stu Brody had been working in circadian rhythms in neurospora for quite some time. Yeah. And he was getting ready to retire, and he had started this class with Michael Gorman, who's in psychology, and who is a mammalian expert. And he was very eager for me to take over his half of the class. And that sounded fine to me. So Michael Gorman and I started working together, and now we taught this class for 15 years, and it grew to be 350 students it grew to and was pushed a little bit by the pandemic to be an online course. So it has now been an online course for a few years. So thousands and thousands of students have gone through this class now over the last 15 years.
But is UC San Diego the first UC to have an undergraduate class in circadian?
As far as I know, yes. And as far as I know, there's nothing like it anywhere in size and for an undergraduate class. I don't even know of a large one in Europe or elsewhere. I mean, 350 students signing up for circadian biology is a lot.
Does Texas A&M has one?
I have to confess, I don't know.
So the point is, in the last 15 years, I guess between you and Michael Gorman, you have trained more number of undergrads at that level, undergraduate level in circadian rhythm than anyone else anywhere.
That's probably true
on this planet.
Yeah, that's probably true. I hadn't quite thought about it like that, but that's probably true. And perhaps, I don't know if I would say more importantly, but the materials that we created with my HHMI professor grant and having a class called the BioClock Studio, the materials that we prepared to use in that big class, we also made publicly available. And some of them have tens of thousands of views around the world. We know that there are circadian labs around the world that when somebody new joins their lab, they have them watch our videos because people tell me this. I occasionally hear from people at companies who say that our tutorials on luciferases, for example, that they'll have their new employees watch to understand what it is their company does.
No, this is for everybody who is watching, because many times we use many terminologies in circadian rhythm and many concepts. And unlike say cell cycle or many things that people are interested in, different disease, you got a lot of information all over the web, but there are very few authentic, validated lecture series like the one you are mentioning that Dr. Susan Golden actually made it possible to have this. She had the vision, it was almost 10, 15 years ago, and I was always curious, why are you putting this on the web? Who is going to watch it? And now it's really gratifying to hear that there are many guest lectures too. So there must have been like 100 or 150 of these BioClock Studio videos floating around that people find extremely useful to understand circadian rhythm concepts.
Yeah, we have a combination of interviews, not as extensive as these you're doing on your podcast, but interviews, tutorials, and some interactive, not exactly games, but simulators or other kinds of interactive activities. So yes, and they're just go to bioclock.ucsd.edu and you can see everything that's there.
And you also started this, or actually Stu Brody was the one who conceptualized the Center for Circadian Biology, and the center has gone through what, 15 years or 17 years of symposium?
Yes, I think that, I think that we've had 15 because the year that Salk had biology of time as a theme for a meeting here, we didn't have one, but I think we've had 15.
Yeah, and in every symposium, this is one of the, I would say boutique symposium on circadian rhythm, because it's relatively small, 350 people max, and it's open to anyone because it's not invitation only. There are many people from all over the world. They register, and also a couple of years when we went online, I guess we had.
Yeah, we had people from many countries that when we had it online. Yeah. Yes.
And that has also become a kind of a template for many other centers to start their own symposia and training materials. So thank you for doing that and doing this for more than almost 14, 15 years.
Well, you're very welcome. I feel like I got a lot out of being director of Center for Circadian Biology. I got to meet people all around the world who maybe might not have paid that much attention to me as just a scientist working on cyanobacteria, but it definitely gave me a lot more exposure to human biology, mammalian biology, teaching the small undergraduate class that I'm teaching right now. I'm doing it almost exclusively on clinical trials, and I feel very comfortable. I feel like I know much more mammalian neurobiology than any other bacteriologist around would know because of this position. So I've learned a lot, and it's been great. And I should say again, thanks to Stu Brody for really. First of all, for conceiving of the center and then pushing us to have the meeting, even our first year when we didn't even have our act together yet. And so we really owe that to Stu, because it's his baby.
Yeah. What is next?
I'm looking at retirement in the not too distant future.
Well, so once I was talking to Sydney Brenner, and I asked Sydney, when are you retiring? Mm-hmm. And then he said, you never retire until you figure out what you want to do next.
Well, I'm not sure that I have met that criterion yet.
But I guess what you have been doing is also exciting, because you're taking a sabbatical, going to different universities, different places, and also trying to teach sometimes other people and train and share your wisdom like you are doing right now.
Yeah, well, that's the fun part. Yeah. Actually, it's pretty much all the fun part, I guess.
Yeah. So thank you so much.
Oh,
it has been wonderful and as I said, you were one of my scientific heroes because of that moment in Scripps when I was a student. And your presentation talking about how you can do anything in cyanobacteria that you can dream of actually inspired me to think, well, maybe within 10 years I can be on that podium. But unfortunately, I have never been on that Scripps podium yet.
They'll have to invite you back. Well,
but you have invited me to teach in your class, yes. So that was also a great moment when you invited me, and I was a guest lecturer in your class. Thank you so much.
It's always a highlight of the class to have you come talk to our students. Yeah. So yeah, always a pleasure to talk to you, Satchin. I really enjoyed it.
Yeah. Thank you so much. And again, hopefully we can have Susan Golden again back after maybe a couple of sabbaticals. Sure. We can reflect on other issues in circadian biology. As you have heard, she is exposed to a lot of neurobiology, clinical trial, human studies, and circadian rhythm from multiple organisms. So maybe next time we can pick her brain on a 30,000 feet view of circadian rhythm and its impact on anything from sustaining oxygen in our planet to human health. Thank you so much.
You're welcome.